Thank you for turning out on a Friday afternoon. It's my great pleasure to welcome Michael Savageau to our company here today. He's actually one of our own, as you'll hear in a minute. Among his many scientific accomplishments, he is one of the founders of metabolic organization theory by various names, covering both steady state and transient state metabolic functions. And among that small number of founders, he's by far the most accomplished in realizing the evolutionary implications of this theory as well. So there's plenty of interest from systems biological perspectives per se, but also from mechanistic studies of evolution. Mike grew up in South Dakota, got his bachelor's degree from the University of Minnesota and his master's from the University of Iowa, and then, for better or for worse, came to mighty Stanford for his PhD. He did postdoctoral work both at UCLA and back here, and was then for many years a mainstay of the Department of Microbiology and Immunology at the University of Michigan, where he rose to a distinguished professorship and sometime chair of that operation. Since 2005, he's been translated to the Department of Medical Engineering at the University of California, Davis, where he again was sometime chair of that organization. He's a fellow of the AAAS, a member of the National Academy's Institute of Medicine, and has been honored in many other ways by diverse institutions around the world. Without further ado, I'll turn things over to Mike, who will talk about genotype-to-phenotype deconstructing complex systems. Welcome, sir.
It's a pleasure to be back here and enjoying some fine weather. What I want to do today is to use the context of the genotype-to-phenotype problem in a way to talk about a new technique that we've developed for what we think of as a way to deconstruct complex systems into more manageable, understandable units that can be put back together without losing information about the whole. Let's see if this works. But first, what's a phenotype? You know, well, you have, of course, definitions like this in the dictionary, observable properties that are determined both by the genetic makeup and the environmental influences. It's true. Of course, there's a famous phenotype you all recognize in genetics. Plants, wonderful. It's not great resolution, but wonderful differences in size and shape and color. You know, those are all phenotypes. In the animal world, lots of differences. Shell patterns, coats in animals, nice eye color contact. And at a certain age, an interesting animal phenotype that some of you might recognize is male pattern baldness. Okay. So on the left, you have someone who, it's hard to see because he doesn't always put his head down on his desk, but if you do, you will see that he has a little spot back here for me. Okay. Now on your right, some would say far right, a guy who's losing his hair from the front. And that's a phenotype we don't quite know what to say about that. But in the microbial world, even, you put down a single cell on a plate and leave it there sometimes for a week, and it starts to dry out and so forth, but you see these beautiful patterns form that are characteristic of the organism and the plate that you put them on, the environment. And even in a culture overnight, you can see differences between smooth colonies and rough colonies that are diagnostic for virulence. So those are phenotypes that microbiologists talk about all the time. But what do you mean by a phenotype of a biochemical system? And here's an example of part of glycolysis that many of you folks spend a lot of time thinking about. And parts of it can be used in either directions for gluconeogenesis or glycolysis. And you could color the reactions that are involved in one process, green, and the other is red. And you might say those are different phenotypes. We're describing them. Here's another. This is a metabolomic study where each dot is a metabolite in the metabolism of an organism, microbe. And they've been exposed to 2 different drugs. You don't have to know the details, but what the red indicates is there's coordinated increase in the levels in the top there that you see in one area, fairly localized, and the bottom it's very diffuse. So obviously a different phenotype in the response to these 2 different drugs. But how do you describe those other than coloring them and talking about green and red and so forth? Another difficult question is how many phenotypes are there? Here's a nice study with a freshwater planaria, and they knocked out every gene with siRNA, and you get all these differences in color and size and shape, all different phenotypes. But that's just some. You know that if you put those genes behind a tunable promoter and vary the levels, you'll see a whole host of other phenotypes come up. So how can you say anything about that? Is even an interesting question. So how do we relate these phenotypes to the genotype? And one of the problems, I think, is that when we talk about the genotype, we know, most people would agree, that there's a pretty good definition of what we mean by genotype in terms of the full sequence, say, of organisms' DNA. So there's a generic description, maybe, of what you might call the genotype. What's the equivalent generic description of a phenotype? Can we come up with one? Well, this is a huge chasm. It's a big black box. And how can we deconstruct, if you like, that box and come up with something that would address some of these questions about kinds of phenotypes, ways to describe them, count them or enumerate them, and so forth. And you could break this into 2 parts, if you like, that combining all the experimental knowledge that you have about how to genotype and environment interact, and you might have some model that describes or summarizes all of your knowledge. But even for that model, what's the phenotypic repertoire of that? Yeah, you can look at all possible combinations of the parameters and try. That's a combinatorial explosion you can't really deal with. So what do we mean by deconstructing something? Well, it's breaking it down into components, yeah, okay, in some ways, but part of the problem is if you break things down into components, if you don't do it in some kind of a structured way that you can put it back together again and have it make sense, then you may just be breaking up functional units, for example, in ways that you'll never be able to put things back together in a reasonable way. So I'm going to propose one method, a technique or strategy for this deconstruction. And I'm going to need these 3 concepts that we kind of agree on, or at least you understand where I'm coming from when I talk about these 3 things. The idea of a system, what I'm going to do to define a phenotype, and then present that in the context of a design space. So, what do I mean by a system? There are lots of definitions. Again, you'll find about a dozen, I should say, or more in the dictionary. But I'm just going to say that the system I'll define as a collection of interacting parts that in some sense constitutes a whole. A little bit vague, but I'm going to say that everything excluded from that collection I'll consider the environment. So that could be many different levels. And so in fact, if the parts that make up the interacting parts also fit that definition, then they're subsystems. In this way, you get a hierarchy of systems that describe nature, if you like. But what are these subsystems? It doesn't always present itself to you in an obvious way by looking at some complex biological organization what the right kinds of subsystems might be. Now, historically, biologists have been very good at putting boundaries around parts of very complex things and understanding them by really judicious uses of controlled experiments where they can kind of nullify the influence of the background they're not interested in. But I'm going to say, as a rule of thumb, not something that's going to always be guaranteed, but say, try and maximize the number of internal interactions to external ones. So this would have a sense of preserving critical functional groupings, because important phenomena cannot adequately be analyzed by just looking at the component parts in some cases. You get emergent properties at certain levels. Such phenomena have to be treated as a whole. So you want to make sure you put boundaries around your subsystems in such a way that you don't violate obvious important interactions. When you do this, there'll still be interactions with the environment, as I just described it, but you'll minimize them. And when you disrupt them to study that subsystem in isolation, you'll tend to identify those, and you can, if you're doing a good experiment, you're mimicking that environment, that proxy environment, if you like, in a laboratory setting. So think of this as a cell, lots of interactions, a few with the environment, and if you just were to count the edges and so forth in here, you'd say, well, there are 33 interactions, 5 external. To just give you kind of a simple lifetime. Okay, how do I represent a biological system? Won't go into the math details, but this is a formalism that we call a generalized mass action representation. It's generalized because the exponents in here don't have to be small integer values as they do in classical chemical mass action kinetics. It turns out that that representation is very generic. And that could be a whole lecture itself, but from many different perspectives, we can say that that's a canonical representation in the sense that you can transform a whole host of nonlinear models into exactly that form. You'll just have to take my word for it. But, you can do that. So it's a very flexible, plastic, whatever you want to call it, generic kind of representation. So what I've said about systems is that they're integration of functional mechanisms governed by physical chemical principles and represented by these generalized mass action kinetic models. Okay, nothing makes sense in biology except in related systems. That's my view. And now we get to the definition of a phenotype. What do we mean by phenotype? Everything that I showed you before, we all agree they're phenotypes. But if you actually pin a biologist down and ask them what they mean by a phenotype, I found lots of different answers. One of my very good friends, who's an eminent geneticist, says mutants are phenotypes. You don't have a phenotype, but you have a mutant. Well, that's one way of looking at it. But I want you to think of an arbitrary biochemical system where I've picked out 3 metabolites. Okay? You've got a certain number of reactions that are producing one of these, and certain reactions that might be utilizing those metabolites. Okay? Now, at any given time, if I took a snapshot of that organism, I would find that there were certain of those fluxes that were dominant, bigger than all the others. Bigger than all the others coming into this pool, so I'd say that one maybe is bigger. Well, this only has 1 going out, so that's not so critical. But here, again, we have 3 coming in and 3 going out, and at a certain instant of time, you might find that this input is larger than these other 2, and that efflux is larger than those other 2. Okay? Mathematically, you know, it'd be very unlikely that you'd find them exactly equal. Okay? So there's 162 possible combinations of these dominant terms. Okay? Well, it turns out that most of those are not feasible. Okay? So we need to test on which combinations would actually be a valid combination of those dominant fluxes. Okay? And that's the way I'm going to define phenotype, as a valid combination of dominant processes. Okay? So what does that mean in this mathematical world, where there's my general description, say, some system with a sum of many influxes and many effluxes, and I'm going to say that there's one of those terms which is dominant, and one of those terms that's dominant. And then you get this equation that I probably spent most of my life thinking about, which has wonderful properties in the sense that you can characterize, it's a nonlinear system, but you can characterize all the fixed points as if it were a linear system. And you get a lot of results out of it. So why does that come about? Well, each dominant process here, if you take the logarithm of that, it represents a set of linear inequalities in logarithmic coordinates. That is, this one has to be bigger than all the other ones in that sum. And in log space, that's just a set of linear inequalities. Similarly, for the dominant terms over here, another set of linear inequalities. And the solution of the fixed points here is also a solution of a linear system. So this is simply linear programming. You can solve a set of linear equations with the linear inequalities, and you get all the boundaries between those combinations of valid dominant processes are linear hyperplanes in this, what I'll call system design space, which I'll say more about in a few moments. But the point is that this gives us some hope that we can scale this to very large systems, because you know that they solve these linear programming problems with thousands and thousands of variables every day, in the airline industry, for example. So this now is a very tractable problem to characterize a dominant, well, I'll call this dominant phenotype. So think of what that does when you find those hyperplanes, is that it ends up giving us a set of qualitatively distinct phenotypes, which we think of as space-filling irregular polytopes in a system design space. So you pick out a set of parameters, and you'll be inside some space like that in a larger dimensional space that we call it system design space. So our definition of a phenotype then is a set or sets of concentrations and fluxes corresponding to a valid combination of dominant processes within a functioning system. A qualitatively distinct phenotype is the characteristic phenotype that exists throughout a valid region, throughout that polytope in design space. And then the phenotypic repertoire is the collection of all of those qualitatively distinct phenotypes. Yeah?
So is this phenotype is for a given set of parameters in that equation is this dominant set of is not time dependent?
Well, you can do time-dependent things, but what I'm talking about today will be the steady-state characteristics, the fixed points, and so forth. But you can move from one region to another. We've done a little of the dynamics of that, but this is still a work in progress. We're still trying to develop tools to extract all the information we can out of these systems and so forth. So there's still a lot to do here. But that's a good point. We do want to look at dynamics. And you'll see a little bit of that, of what we deduce from the fixed points in a moment, probably. So from that kind of a definition, I think now I've got, for me at least, a comparable set of concepts for relating genotype and phenotype. The genotype is the collection of all the genes in the system, and the phenotypic repertoire is the collection of all the qualitatively distinct phenotypes of that system. Okay, the last thing is design, which when I was a student was a dirty word. And it was a dirty word because of fundamentalists who were trying to discount evolution, and a certain group of evolutionary biologists who were dreaming up just those stories and so forth. So when I came to publish this book and wanted to talk about function and design, I got some good advice from one of my advisors here at Stanford who said, yeah, use that word. It's really a great descriptive word for what you're talking about, and don't let other people co-opt it for bad purposes. And so I stuck with it. But nowadays, design and design principles and the search for design principles in biology is a big cottage industry now. There's a lot of interest, conferences, textbooks being written, and so forth. So let's get to the main point here. I want to put this notion of phenotype and genotype and environment together in a way that I can relate them all. So I'm going to do it by means of a toy example that I'll go through 3 different stages of kind of expanding it, but real simple so you can get the main ideas. So the system here is a couple of reactions that are involved in relating the 2 different cyclic forms of glucose, and there's an acyclic intermediate. It's been well studied probably 100 years ago. But here's the way I think about that as a system, if you like. So here's the environment. In the laboratory, I can determine the concentration of S and the concentration of P, and I can set up different thermodynamic potentials between those by adjusting the concentrations. But everything within this box is going to be my system. It's a very simple system with one dependent variable, and we can think of these parameters as being genotypically determined, if you like. I'll come back and clarify that in a moment, but that's a very simple system, and that has 4 dominant phenotypes, 4 dominant ways of combining the fluxes, if you like. And here's the equation that goes with it, and there's a solution, and here are the conditions where it's valid. And what you see for the conditions here is this is a genotypic constraint, if you like, on the kinetic parameters. And over here is the environmental constraint, the displacement from thermodynamic equilibrium. And that combination defines each of these phenotypes. Okay? So when you plot that out, here is the environmental variable. And we often try and do this when we're trying to study a system where we have some interest in an environmental variable and looking at some genetic determinant. So here is the displacement from thermodynamic equilibrium. So at 0 here, it means that we're setting at equilibrium. So you can see by these little arrows here that every point in this space on the right here is a flux to the right, and every point on the left is a flux to the left. And they're just these 4 regions. Now, what does that mean in terms of kind of your intuition of what those phenotypes are? Well, here's what it is. In this region up on top, region 1, it says that the first reaction is a rapid equilibrium, quasi-equilibrium condition. And the flux is either to the right here, by this essentially unidirectional flux, or to the left. symmetrically disposed down here is the case where the second reaction is in rapid equilibrium. And then, of course, in these slices in here, you have both reactions being driven hard in one direction or the other. And that's really all the behavior that you can expect from this simple system. And you can count them. There are only 4. And now here is an example of what you can do in the z direction. You can plot any characteristic of that phenotype in the z direction that you're interested in. So in this case, the simple system would just plot the normalized concentration of the intermediate, and you get this thing. Now, when I ask people, if you look at that, how would you differentiate the phenotypes in this? And almost everybody would say something like that. Well, there's a blue region and a red region and a green region. And they'd draw some boundary that might look like that. Well, here's the solutions based on those dominant cases. And you can see that they're a little different. These have a little sharper corners and so forth, but pretty much they look pretty similar qualitatively. But the main thing is that we're interested in is not the precise accuracy in some of these corners, but rather where the boundaries are telling us. These come out of the actual mathematics. They're defined, they're not arbitrary. And those tell you something interesting, because now instead of having 2 different phenotypes up here, there's 1. And that phenotype is only dependent on the environment. Okay? There's no genetic influence on things up here. In this little triangle here, it's only dependent on the genotype. The environment isn't making any difference at all up here. And in that corner, you're getting a combination of both genotype and environment are interacting to give you the behavior in here, and there's this null region, which is the same. Now, if you take the difference between the actual solution and in these dominant cases, for the most part, they're essentially identical. Where you see the error is right at the boundary, because there is no dominance at the boundary by definition. They're equal there. So that's where you see the biggest difference or error. So we're not interested in that error so much as the precise definitions of the boundaries that are helping us define the phenotypes. Okay, so suppose that we wanted to characterize the distinct phenotypes of this chemical system, say, in a fixed environment. So just looking at the differences in the genotypically characterized things. How might we want to look at that? Well, we could look at the concentration. I just did that. But we also could talk about the net flux through here, through this. And we might talk about something called toxicity. That is, for a given flux through the system, looking at the concentration of this intermediate, if it's elevated and that is toxic, then that would be a disadvantage. You'd like to lower that concentration for a given flux, say, through it. Yeah.
So I'm just curious, where does the genotype come in here? Is it embedded in the K1F, the forward example?
I'm speaking of these as genotypically determined parameters. These are...
So the Ks are genetically determined parameters.
Yeah.
So your model doesn't conceive like of the transcriptional process or the
not for this. Not for this. I'm trying to look at the simplest system at just a metabolic level.
Okay. But your phenotype, you measure the phenotype via the metabolite concentration, not the fluxes of the...
No, the phenotypes are defined by these dominant processes.
So it's like basically the extraction point of qualitatively which state space we end up in, determined by the
well, you ask what determines those boundaries, and there are these parameters that will call for now genotypically determined parameters, and the environment, the relative concentration of substrate product. Okay. So, anyways, these are some things you could talk about in this very simple system. And so again, we have the design space, and we could say, let's fix the environment with a particular combination of substrate and product and the thermodynamic parameters. And we'd be looking at a slice through here where we would just be varying the kinetic parameters. And what you see from that slice, and you can take those parameters and put them on the axes, it's kind of the same 3 things if you go up through here. This red region corresponds to this, the colors change, but it's this region, and the blue is this. So we're sampling everything along this line for all the different genetic parameters in this space. So it's a slice through our design space. Okay, so on the z direction, I can put different things. I can put the concentration. And what you see is if you vary these genetically determined parameters, that the concentration goes up and gets very high over on this side. And you can also look at the net flux, and you get something that looks like this. And here's that toxicity I defined as the concentration that will rise for a given flux. So you could think of being up here. I'm sorry, this resolution isn't any better, but my slides are better than this. Must be my setting for this projector. But anyway, you could suppose you're at this flux, and for that given flux, if that's what you had to achieve in your physiological setting, you could vary it this way or this way, and you'd have the same flux. So that's how you can move in this particular design space with a simple system. You have, if I want you to have that flux, you can move up or down this way. But what you see is that if you move up along here, you're decreasing the toxicity. Okay? But if you move in this direction, it stays about the same. Okay? That's about as much as I can get out of that simple system. But now suppose, and this is where my use of the term genetically determined parameters is really not accurate. That was a chemical system. I didn't have any choice about those parameters. That's determined by the chemistry. And if you like, that was the chemical evolution of the universe. But now let's talk about the biological situation where those reactions are catalyzed by enzymes, which do have a biological influence that we can talk about. And I'm going to talk about this same system, but now these are Michaelis-Menten kinds of reactions, but operating in the linear domain. So this is a linear system, and in fact, if you group these parameters together, they're like the K's in the chemical system. But now these are biologically evolvable parameters. Okay? In fact, yes.
So where does the environmental factors come?
Environment is the substrate product. That's what I control as an experimentalist.
Okay.
Okay? And you've got these kinetic parameters that are fixed.
And the kinetic parameters are from the...
I'm sorry, these are thermodynamic parameters that are fixed. In fact, there are 4 relevant parameters for each of those reactions, but only 3 of them are independent because there's a constraint by the thermodynamics that the other parameter has to satisfy. So for each of these, we've got 3, but in this particular context, I'm looking at it when they're far from saturation. So the idea is to kind of mimic the chemical system. But what's different here now is that we can again talk about substrate level, and here we can talk about the net flux. But we can also talk about what we might think of as a cost. You know, how much energy it takes to make the enzymes for this pathway. And we could just take as a crude measure the sum of the concentrations of the 2 enzymes, which I'm representing here by the Remaxes. And again, we have something like toxicity we can talk about, but we can also talk about the efficiency. You want a given flux to the system, how much are you willing to pay? You can move in design space and find the same flux, but at a cheaper cost, there'll be some selective pressure to go in that direction, or to have survivors in that direction. So you get a little bit more information here. Again, this looks very similar to what we saw before. This looks exactly the same. This looks very similar. But now we've got these other 2 things here. The cost, as you look at the levels of the 2 enzymes, and here's the efficiency. So if you were sitting here, again, at this point, and you wanted to maintain this flux, you're on this isotherm here, you could say, well, if I move in this direction, what happens? Well, the toxicity doesn't change, but you would lose efficiency. Okay, it would cost you much more to maintain that flux with value over here. On the other hand, if you move upward, you're still maintaining exactly the same flux. In this case, the toxicity goes down, so that's an improvement. But again, the efficiency goes down here, which is not so good. So there's a compromise or a trade-off between these 2 effects. So you might decrease the toxicity, but lose a little efficiency. So you start to see that there's a multi-objective function going on here if you were thinking of an optimization process.
So essentially, this concentration, flux, cost, efficiency, are just objective functions you try to see how the system maximizes or minimizes energies.
Well, this is how it would, given all these different things. So if you were, say doing an evolution experiments in a computer, you could throw down dots that would have different combinations of things in this plot, and you could expect these kinds of
But is it possible to infer the objective function from your data?
Well, no. I mean, I'm setting up what I'm calling cost and what I call efficiency and toxicity. You have to define something. It's not going to come up with criteria de novo. if I misunderstood what you're saying.
Yeah.
It's not telling me what the criteria are.
Because from the perspective of the bacteria itself, we do not know what it's trying to optimize, right?
In a larger sense, I would agree with you. But I think that toy system, this is the system. And what choices do you have? Okay, you've got an environment, and you've got some genetic determinants, and given any combination of those, this is the behavior. And it's a way of teasing that behavior apart. And what you see, in some sense, is that you want to be on the edge of this phenotype in here. That seems to be an efficient, low toxicity, you know, and you can get whatever flux you want by moving along that boundary. Okay, so go one step further and look at this in the unrestricted domain, because in fact these processes can saturate. Right? Not linear anymore. So this is a nonlinear system, and we can still have these criteria we could talk about. And if you go ahead and do the same thing, what you see is design space gets more complicated. And sort of this box in here is much like that linear behavior. It's part of this. What's happening out here is you're saturating one or the other of those enzymes, and you get different behaviors. Again, you might want to move along the diagonal here to increase the flux, and you could keep increasing to a certain point, but once it saturates, you keep moving out here, and you don't get any more flux. Again, with a given flux, if you were trying to maintain it, you can move in this direction, and toxicity stays about the same, but again, efficiency would drop. If you move up here, toxicity goes down, but you also move out from the optimum, which is along here. But you see, it started off kind of in this trough, which is then the optimum when you're a linear system, and with a nonlinear system, it turns a corner, and at some point, the best you could probably do is move along this line here, where you maintain low toxicity, high efficiency, and some high flux out here. Okay, so that's the simple toy system. So let me turn to a biological example, which may give you a little better appreciation for what we do in a context where we don't really know all of the details in some sense, although in this case we do know an awful lot. So you all know probably phage lambda, and it has this two different phases of the life cycle. That a lot of attention has been played to this decision here about whether when it's infected with this, the cell is infected with this phage, whether it goes down this pathway where it produces more and more copies of itself, lyses the cell and releases the phage to go on and infect other cells, or whether it inserts its DNA into the host chromosome and gets replicated passively along with the host. Okay, so there's a decision made at some point in here. A lot of people have talked about that. There's another one here that if you are integrated into the host chromosome and the host is damaged in some ways, there's a decision, if you like, made about whether you think the host can survive that damage by repairing the DNA and so forth, or whether you should induce, get out of that cell, reproduce, make copies, and try and infect another cell. So this decision turns out to be fairly simple compared to this one in some respects. But here are the known regulatory interactions. I won't go through all these, but in lysogenic growth, there are very few functions being expressed. Basically, it's quiescent in the cell, and it's basically making a C1 repressor. That repressor represses both the right and left arms. It keeps all the lytic functions turned off, but it's also an activator of its own transcription and represses at higher concentrations. And so that's the little gene circuit that we could think about in terms of looking at induction. And the molecular details have been studied exhaustively, and we know all kinds of kinetics of the binding of each of these little sites and so forth. And you can put that all in a model. Okay? And when you, in fact, combine those equations, you get a nonlinear model that looks like this. It's rational functions. It's even got some radicals in here. And it's nonlinear, but we can recast that equation into what I call that generalized mass action form by defining a couple of new variables. So if you define the denominators in these expressions as a couple of variables, now this all has that generalized mass action form. And now you can actually count how many possible combinations of dominant processes there are, because it's choosing one from this side, one from this side, one from this side, one from this, one from this one. So you can easily go through this, and there are 54 possible combinations. possible combinations, possible phenotypes. Okay? So that's what the design space looks like for this when you put all that information together. There are actually 10 that are realized. There are 3 that are lytic-like, and there are 4 that are lysogenic-like, and there are 3 in here which are hysteretic. Okay? So each one of those regions we can characterize, just as I showed you for that simple model, in great detail. We can look at local robustness, we can look at all kinds of amplification factors, stability, response times, and so forth. When you do that and compare them, the best lytic region is over here. When you ask the same question about the lysogenic region, what is the best, again by a number of different criteria, it turns out to be this region, and in fact these dots represent the experimental values where the system actually operates. Okay? So here again on the horizontal axis is our environmental variable. It's DNA damage, it's radiation, whatever you want to think of it. And this is one of the genetically determined parameters, which is the affinity of the C1 protein for its own activation. So when you experience different amounts of damage, you're moving along a line like this through this space. And these 3 regions are actually, well, take region 7. There are actually 3 regions in that region. Because the boundary of region 1 goes down to the lower part of 7, and the upper boundary of 11 goes over that region, and then there's a third. So there are 3 steady states in this region, and that's the signature for hysteresis. Okay? And they exist through this band here. So here's a hysteretic region which you can look at in one sense as protecting you from inappropriate induction. That is, if there was some noise, but you could get back and the organism corrects that damage through its repair mechanisms, you don't want to start the whole lytic process. But once you cross that boundary, it's hard to reverse it. You've got to go back to the other boundary where you can switch it back into the lysogenic mode. So there's a buffer there created by that hysteretic zone. Now, if you also put a little bit of noise in the system, what you'll find is that you can get a biphasic population in here, where there'll be some that'll be lysine and some that won't be, and that's hedging your bets. And the wider that region is, the more difference you make these biphasic populations. But the other really interesting thing are these dotted lines in here. Those dotted lines are determined by these particular boundaries in the design space, and they're determined by all the parameters in the model. Okay? And what it says is that if you were to change this particular parameter to move the plot point up here, you would be locked into this lytic mode. You couldn't get back. You would be like a T4 virus, where you just grow lytically from one cell to another. Conversely, if you change this parameter to put this dot down here, so it's operating in this region, you're always going to be a licensed, and you'll die with that host when it dies. You can't get out. Okay? So in order to maintain that biphasic lifestyle of phage lambda, it's got to have parameters which keeps this operation within that band between those 2 dotted lines. Now, I mentioned in terms of this one parameter that can do that, but in fact, many of the parameters can do that. What it means is that you change a parameter such that that boundary moves down and leaves the point above it. Or you change one of the parameters that influences this boundary to move it up, and then that boundary is below it. So there are lots of different ways to put you outside that band, but if you do, you're not going to be a lambda virus. Okay, just to summarize some of these points then, that model actually has 15 parameters in it, and we found a way to sort of compress that information into this 2D space. And we predicted the behavior in these different regions, and there are 3 types. There's the lytic and the lysogenic, and then there's this hysteretic behavior in here. And we've sort of identified a couple of design principles that you can get 2 kinds of pathology. You can not cycle properly if you get outside this band, or you could have some inappropriate switching here if you didn't have a good enough hysteretic buffer. And there are some other properties like the asymmetry and switching times. It's much faster at induction and getting out of an alien host than it is when it goes in and integrates. And you can think anthropomorphically about that if you like, and that, if the ship's going down, you want to get out fast. So it kind of makes sense. Now, the other thing that I haven't talked about yet, which I said you could measure the robustness in each of those regions. And robustness, in most cases, is often talked about as a local perturbation, parameter insensitivity, if you like, because you can always calculate those for most systems. But what do you mean by robustness to large changes? Changes that would completely alter a phenotype or something like that. Well, we think that this kind of view is giving us a notion of what we call global tolerance, to kind of distinguish it from robustness. That says if I'm operating here, how much do I have to change that parameter before I get to a different phenotype? In some cases, I can change it enormously, so it's very tolerant to changes in some of these parameters. And in some other parameters, they'll be more limited. But you're basically saying, what's the distance between the boundary to another phenotype from where it's normally operating? That's a measure of tolerance, global tolerance, big changes that would alter the phenotype. And what we see when we do that is that all these tolerances are very large. These are fold changes. They're not percentage variations in parameters in most cases. And in the lambda example, there is a lot of good experimental evidence that supports that notion of global tolerance in John Little's work at the University of Arizona. He's basically taken all the parts of this system and rearranged them in different ways. There are these 3 different operator sites, and he's exchanged all their positions, and their strengths different. And he's even taken entire control modules and taken out the C1 repressor and its recognition sites, put in a different kind of repressor with their recognition sites. And for the most part, it still behaves like a normal phage lambda. So it's very robust to point changes and large changes in the whole architecture of that system in many respects. So, summary is that I think of biological systems as functionally integrated mechanisms governed by physical chemical processes, represented by genomes, mass action kinetics. Phenotypic repertoire of a system is an unambiguous concept. I think it's comparable to the genetic repertoire. And the system design space is a way of relating these. The phenotypes are associated with regions in that space. You get a bound on the number of possible qualitatively distinct phenotypes. You can begin to compare the fitness of all these phenotypes because you can analyze each with very standard mathematics. You've got precisely defined boundaries between these phenotypes, and this novel definition of global tolerance related to the distance to those boundaries. And in some cases, we've seen how it facilitates the identification of what I think of as a design principle. These are some of the folks who contributed to this, some of our collaborators. This is something that comes from my wife, who's an artist, and she talks about every true artist has been inspired by the beauty of line and color, and so forth. This is a quote from Mondrian, and he's famous for these colored rectangles and things, and I looked at that and said, well, he's very rigid because these are all straight vertical or straight horizontal lines, and ours are all skewed and so forth. And this is just a collection of some random cases. But these are like a fingerprint. Every system we've looked at has a different design space. And so I think this tells you a lot about phage systems. Now, one more thing is when I showed you the simple system and I showed you one slice through this fixed environment and what variations in the genotype could do. And if you allow for all combinations of environment and genotype, there in fact are 32 possible quantitative phenotypes. And so I can show you just very quickly a couple more slices. They look a little different. This kind of has a little wedgy thing. And here's the concentrations that kind of go with this design space. Here's another one. And you get this little trapezoid in here with low concentrations. You get this square in here, some concentrations varying around that. This is the one I find most interesting in many ways, is you've got this triangle and this thing in here. And look at this bizarre behavior. Concentration is very high. When this is low or this is high, this is pretty high. And in between, you have these weird triangles and things. Fingerprints, right? Okay. That's about all I'm saying. Thank you for your attention. This light hour on Friday afternoon. Thank you.
Please direct your questions to the speaker.
So, from those parameters, has it been experimentally shown that you can push the bacteria towards a lysogenic stage? As in, did you experimentally verify that? from the parameters that if I knock down certain genes, you push it towards, like certain enzyme, you push it towards a certain phase.
There are a lot of mutants. This is one of the best studied molecular systems that you can probably find. And there are a lot of mutants, in fact, that are in fact identified. Those regulatory circuits were identified by mutants which knock out those regulatory interactions. And so yes, there are mutants which become lytic.
But from the prediction, can you?
They haven't measured all those parameters. What we do have is the parameters for the wild type, and we have. I've talked to Sean a little bit. He's trying to measure some of those in all of his mutants. But you know, most microbiologists in that era were not giving you numbers. They were all in on mutants almost, where they would look for a clear zone, which meant that it was a lytic mutant, that it only would lyse cells and not lysogenous.
So those are all bulk equations. What about, like, if you go down to very low concentrations, then you go to random interaction? I don't think those equations apply, right? Those are bulk chemistry equations.
These are biochemistry.
Yeah, so if you go down to very low concentrations, it's more stochastic, and it won't be described by the differential equations anymore.
Well, actually, you can take this down. I mean, when you look at most of the stochastic simulations, you still have to have an underlying structure. You don't just simulate random things. But if you ask, will the mean behavior of the stochastic system agree with the deterministic? No, it doesn't always agree. In fact, we've got a paper publishing right now that does just that. When you put noise into the system with low numbers, you get a lot of understanding from the deterministic model, even though that, you know, it's some kind of an average, but it's not even the exact mean of the stochastic process. But you get information. When you put the stochastics on top of that, you get insight. For example, the width of these hysteretic regions, that determines you know, how, in the paper we're talking about, whether you get persister cells. These are microbes that are not growing and they're resistant to drugs. So you get a population which has got the same genotype, but it's gotten into it's a biphasic population now, with some cells that are hardly growing and others that are growing. And to get the right combinations of having enough persisters around so that they're hedging their bets against difficult times, and that, when you put the noise in the system, you can actually predict that.
So you can put a boundary just looking at the overall statistics of the population of genotypes. You can guess at the level of noise that's present.
Well, you can, yes, I mean, if you can see from the width of some of those regions, if you put a noise process in there which is too large, you just swamp out everything, and you just get crazy random process. But if you put in realistic amounts of noise, you know, the 10, 20% kind of thing, what you find is that makes use of that together with the hysteresis to give you some bet hedging. No.
Did I understand you correctly to think that different regions would then have different sensitivity to a given amount of noise?
Yes. I mean, sensitivity to variations in any parameter, whether it's an environmental one or any of these genetic properties that you could think of as being noise as well. So I think I distinguish between, for the purposes of this, the local variation, and I think of that as the robustness issue. That is the way many people have described it. If you think of robustness in the more general sense of any kind of change that it can withstand, then I'm trying to differentiate that local kind of noise and variation from the big changes that would change a phenotype, which we'll call tolerance.
And with those, would that tolerance change only as you cross boundaries into different regions, or might the resistance to changes also be gradient within regions?
Well, the local robustness within a region will vary. To the small variations. But the way we're defining this tolerance is simply take where you are in that space, in that region, and ask how much you have to change it to cross a boundary to another region. And so once you cross and you're in another region, then you have a whole new set of tolerances. Once you're in that region again, how far are you from the boundaries in that phenotype? How tolerant are you there? And what's interesting is the systems where we have some pretty good numbers, experimentally determined numbers, that they tend to reside in what we analytically come up with as the best phenotype. in many cases, they're also far away from the boundaries to a really bad phenotype. So it's as if not only do they have local robustness, but they've become very tolerant to at least the bad regions.
So natural buffers.
Yeah, I didn't bring that data, for example, but if you look at glucose 6-phosphate dehydrogenase, I think is the enzyme that's been most studied in humans because it's involved in resistance to malaria in some people. And there are something like 300 mutants in that, and 100 or more have been well characterized by standard protocols by the World Health Organization. So a lot of numbers on that. The design space for the oxygen stress system in erythrocytes, you do this design space, it's got three regions. It's got a good region, according to a number of criteria that I'm going to time to go into, but you say there's one that's kind of a moderate, and there's one that's really bad. And the mutants that have chronic disease are in this bad region. I should say in the intermediate region, let's call it bad. The really bad, you don't find any mutants in there. It's like they're lethal. And then you have cases where the parameters are such that they're in the good region, but they're close to the boundary. These are patients that can be environmentally induced to cross the boundary in a sense. They're sensitive to fava beans for some reason. I don't know why that's an issue. That's a stress somehow. But or certain drugs, and so when they can cross into this where their red cells start lysing, and that's a phenotype that's kind of variable depending on whether they're under stress or not. And those ones are close to the boundary, and you can relate that stress to how the system responds at those points go across the boundary.
Yeah, so I'm a little bit confused about how you take something that has a sort of 15-dimensional parameter space and reduce it to 2 dimensions.
Well, it's in part by in this case, making use of the experimentally determined parameters. So
that's the thing. I mean, like how do you determine these rate constants and whatnot in vivo?
Well, let me give you two parts of the answer. One of them is that in this case, with the lambda case, with that model, you can actually define dimensionless groups, and that's part of the answer. So all of the information is in that 2D space, but the boundaries are going to be a function of all
There
of
has to be more than 2 dimensional groups. I mean, so many.
It's a slice through that n-dimensional or whatever dimensional space it is. But the point is, when you take that slice, you get boundaries that are functions of the other parameters. So if you were to move in and out, you would see those boundaries, and we can actually put a constellation of parameters on the position of that boundary. So in that sense, we can still say something about the 15 parameters in that 2D space. But the second part is even more interesting, and that is you can take a system where you don't know any of the parameters, okay? And you can do vertex enumeration and ask, is there a consistent combination of dominant processes? Is there a region? And you can identify all the regions without knowing any of the parameters. And you can in fact find at least one set of parameters that will locate you in each of those phenotypic regions. That's kind of amazing. It doesn't tell you that that's the set of parameters you'd be most interested in, but you can find a set of parameters that will put you in each of the viable phenotypes. And then you could play around with the parameters, maybe of a given system that are different from that, but in that same phenotypic region. So it does, this thing is at least, I think of these space-filling polytopes, and you can get yourself into each one of those, meaning that you can take a slice that'll visualize the behavior when you're in that phenotype, which is pretty amazing. And it's all linear mathematics. I don't know if that helps answer your question, but
I still don't think you can figure out where the actual critter is, though.
well, I'm not saying I can tell you where the critter is without knowing anything. But what I can tell you is if you've got a model where you don't know the parameters, but you're pretty confident that that's capturing the, in a symbolic sense, the interactions, you can produce a design space and find at least one set of parameters for each of the phenotypes of that model.
But like when you start making up these mutants, or you have all this set of mutants, you don't really know that you have to sort of fix a bunch of other dimensionless numbers, and then you take it,
sort of 2D slice and whatever dimensional space. And you have these regions. But if you start making mutants, I mean, you take all mutants, you don't really know. I mean, mutants are very unlikely to stay constrained on that particular 2D slice.
Absolutely. No, but what we can do though is in that slice, I can determine the tolerance to the next regions.
Sure.
So I can, if you're in some region and you take a mutation.
As long as you're staying in that 2D.
No, even in the 3D. The tolerance, once you have an operating point, you can determine the distance to boundaries. Now that in the 2D thing that you can actually look at it and say, okay, it's that many inches or whatever. But what you're not seeing is you can go in these other directions in that design space and calculate what the value is of the boundary, which will take you to another phenotype. So you can calculate, for example, what I just told you about always being able to find one point in each of these phenotypic regions without knowing any of the parameters. You can now take that point and ask, well, what is the tolerance if I move to any other nearby phenotype? And so you can see if that one point is one that puts you at the edge of the phenotype or in the middle or wherever it is. But, you know, that's still not going to tell you what your system is. I don't know any way to get at those numbers except measuring them. And that's always a hard part.
I'm assuming those parameters will be like protein affinity parameters. So you can people actually did experiments to calculate those parameters.
Well, in the lambda case, yes, that's where most of those numbers came from. They really have
So you
those
can actually
affinities.
estimate
Yeah.
Okay, if there are